Well, welcome back, everybody. Before we get started with Hannah's iNaturalist workshop, I'd like to introduce John Dunn again, who had a couple of announcements to make.
Thank you, John. With apologies, as going back through my notes, I just wanted to make a correction and a couple of additions. One, Kurt Leuschner is certainly on our board. He doesn't vote unless it's a tie, but nevertheless, he's actively contributes and he heads up our tours committee. I never really described Liga's role as recording secretary, but for your information, she translates our conversations that are taped in the board meetings and tries to make sense of what we say in a transcript, difficult when we're talking over each other and arguing. So Liga deserves thanks whenever you see her. I should have added on the monographs, mentioned the ones we've published, which include Trends and Traditions and Species of Special Concern and the Rare Birds of California and the LeConte's Thrasher. But we also have ready to be submitted or nearly ready the Birds of Inyo County by Tom and Joe Heindel, which is being squired through by our previous editor on special publications, Ken Abel, and 3 more are at least in the embryonic stages of consideration. And finally, just to deal with it here, there was a question submitted later on Tule Goose. Could they be at Lake eBird, which is close to Summer Lake, only 25 miles away or so, in Lake County, Oregon? And the answer is absolutely. I think this fall, wherever there's water, that will be determine the distribution of Tule Goose and no doubt a host of other waterfowl. I was alarmed when I looked at Goose Lake, which straddles the Oregon-California border. It was bone dry as of spring. And that concludes my comments. Sorry for the interruption. Thank you, John.
Thanks, John. I'm delighted to introduce Hannah Floyd, who will be conducting this iNaturalist workshop. Many of you saw Hannah's presentation earlier on following mold in individual birds over time. And she is going to be high school junior in the fall. She's a native of Colorado and interested in a wide variety of subjects having to do with ecology, natural history, math, and so on. She enjoys photography, and she's written a number of papers in birding magazines. And I'm very excited about this topic. I think I have many birding friends who are interested in so many aspects of natural history. And as the momentum builds with people contributing data to community science, I think iNaturalist is filling a need that is very strong. And so I'm really happy to have Hannah talk to us about this today. Thanks, Hannah.
Thank you so much, John. And thank you everyone for being here tonight. So first thing I'm going to do is share my screen. So hang on just one moment. And let me pull up this presentation and then present it. Okay, so I just want to make sure that y'all can see a presentation that doesn't show any speaker notes or anything, but that looks, can we see the presentation? Okay.
Yes, all is good.
Okay, perfect. So most, as you all probably know, we are here tonight to talk about iNaturalist. This workshop is geared towards beginners mainly. However, if you are an old timer or expert at iNaturalist, there are certainly things here that may be useful to you as well. So moving right along, I want to remind you guys to please use the Q&A feature or chat feature in Zoom throughout my presentation. I do have a designated question and answer time at the end. However, throughout the presentation, please do, if you have any questions or comments about something that I said, put that in the chat. I don't think that I will be able to see the chat, but Jen, if and when those comments come in, if you could just tell me when I'm done with the slide, because I would like to answer those as live as possible. And then the other thing, this is an hour-and-a-half long presentation, which is pretty long. So we will be taking a brief 5-minute break at 5:45 p.m. So 6:45 p.m. my time here in Colorado. But please do be back at 5:50 because we'll start promptly after. So if you need to go to the bathroom, grab a glass of water, I don't know, grab a bag of potato chips or something, call your grandma, do that in that 5-minute period, and then please do be back at 5:50. All right. So before we get into it, I want to introduce myself a little bit. My name is Hannah Floyd. I am a junior in high school here in Boulder, Colorado. And as you guys know, I will be talking about iNaturalist tonight. I got into iNaturalist. I had been dabbling with iNaturalist for a little bit since about May of 2019, but I seriously got into it right after the pandemic started in March of 2020. I didn't really have school, so to speak. I had about 20 minutes of school a day, which doesn't really count, and I had a lot of free time. I wanted to have some way to spend some time outside, and I'm fortunate enough to live right next to a couple duck ponds, a wetland marsh, and some lakes that are within walking distance. So I began to spend a lot of time there in those beginning chilly March days in the beginning of the pandemic. I started observing all of the usual winter ducks, all of the pelicans, all the great blue herons, all that kind of stuff. And then by the end of this summer, which is what this article is about, I was able to identify a ton of different stuff that I hadn't been able to do as well at the beginning of the pandemic. So that purple flower, tiny, I knew what it was, the Baird sandpiper, and then also that brown butterfly, which was actually a state first for Colorado on iNaturalist. However, this talk is not really going to be about my journey, so to speak, of using iNaturalist. You can read some of my other articles. This one was published in the December issue of ABA's, or American Birding Association's Birding Magazine, and I have done other articles, but like I said, this is not what we're going to be talking about tonight. Tonight, we are going to be talking specifically about how to use iNaturalist. And I'm going to do that by way of taking you guys on sort of a virtual tour of a little walk that I did with my dad exactly a week ago today, of the wetland marshes and duck ponds around our house, and show you some of the organisms that we saw and how we uploaded them all to iNaturalist. So you can see there that my dad is also credited there at the bottom. Some of the media will be by him, and I will be sure to point that out when we get to that part of the presentation. Okay. So moving right along, I know that this presentation is about iNaturalist, but I first want to start talking about eBird. So I'm sure that most of you, if not all of you, use eBird, know what it is. You probably use it all the time when you go out birding. I know I do, my dad does. And I want you to note that, as usual, there is a location, there's a date, there's a time, it has the mileage that we had, there is a checklist comment about some of the general things that we did. There is media, and there are comments for each individual entry. And then one thing that I also want to note here is that this checklist, which was created by me, is shared with my dad. This is an important distinction from iNaturalist. On iNaturalist, you actually aren't able to share observations with other people. but you can do that on eBird. So you can see a lot of the normal expected summer birds here. We have, hummingbirds that pass through here at the end of the summer. If we continue down the checklist, again, more hummingbirds, great blue herons, expected summer birds, nothing super crazy. We're gonna keep going, and there are still more just, expected, nothing super crazy, nothing out of the ordinary. And we have, again, more media comments for each entry. And then our checklist continues. It was a pretty short trip, but we had a lot of stuff. And the thing that I want to point out to you on this slide is the gray catbird entry, which has 3 different kinds of media included in its entry. So there is a photo, which is the left most entry, and then there is an audio file spectrogram in the middle, and then on the right is a video of the gray catbird frolicking around in a water feature near the bird feeders. So keep that in mind for later. We're going to come back to the fact that eBird allows you to have video, audio, and photos all within the same entry. Okay. And then finally, to wrap up our checklist, we have 2 last entries, and I also want you to keep in mind the common grackle, because that bird will be coming back to us in the end in a sort of unexpected way. Okay, we are now finally going to get to the iNaturalist portion of this presentation. We are going to start with a black-chinned hummingbird. We have a lot of hummingbirds that come through, or not a lot, but we have hummingbirds that come through here in Colorado in late summer, and you can distinguish this from a similar looking hummingbird, the ruby-throated hummingbird, by the brown cap in a ruby-throated. That is going to be a vibrant green, glisteningly green cap, and then also the inner primary feathers here are rounded. Again, in a ruby-throated hummingbird, those are going to be quite pointy. And something to note here is that, so ruby-throated and black-chinned hummingbirds, they are the only 2 species in the genus Archilochus. And we also get both of them regularly at the end of the summer. They pass by through here in Colorado. So with this photo, and so now you're like, okay, well, I have a photo, so what do I do now? How do I upload it to iNaturalist? So the first thing that you're going to do is you're going to get to this screen when you hit upload. So you're going to notice that I have filled in 2 of the boxes. That is the location and the date and time. Now, it is very, very, very important that you fill in these 2 fields. Without those 2 fields in, without those 2 parameters in, when the iNat algorithm goes in to try to identify whatever you're looking at, it uses those 2 parameters. So it is so important to put those in, because otherwise it's not going to give you a precise identification of what you're looking at. So I put in the location, Lafayette, Colorado, and you can make that as broad or as specific as you want it to be. And then I put in the date and time, August 21st, and 11:43 in the morning. The final step in this stage is to click on the species name. And you can see that it was spot on. As we were talking about just a couple slides ago, there are only 2 species in the genus Archilochus, the black-chinned and the ruby-throated hummingbird. So it got those both right. The black-chinned hummingbird is the top suggestion, which it actually is. So that is also accurate. So you click on black-chinned hummingbird, it goes in that species name, and then you upload it to iNaturalist. Now we're going to look at a different bird from this trip. A black-capped chickadee, which was a few feet away at a different feeder near the hummingbirds. So again, we are going to plug it into the iNat database. We're going to, again, put in those locations, Lafayette, Colorado, and then you're going to put in your date and your time. And like I said earlier, that's really important that you remember to do that. Otherwise, it's not going to be able to give you an accurate sense of what you're looking at. So with this one, when you click on species name, you can see that it gives me the correct identification of the bird I was looking at. It gave me black-capped chickadee for the top suggestion for my identification. And I want to draw your attention to another feature of this suggestion box that we didn't talk about in the previous example with the black-chinned hummingbird. And that is the include suggestions not seen nearby tab. So you can see with the current image, all of the species it suggested, the black-capped chickadee, the mountain chickadee, and the pygmy nuthatch, it says visually similar, so the bird looks the same, and seen nearby. So there was other observations nearby of this species. So, and what happens when you click on include suggestions not seen nearby, which is something that I don't recommend doing, because you'll see what happens. When I click on that, it gives me species that could look similar, but aren't necessarily seen nearby. The Carolina chickadee, for example, is an eastern bird, has no record in Colorado, so that is obviously not correct. The cinnamon rumped seed eater, which is something that I wasn't super familiar with before this presentation, or before this this observation that I put in. But when I looked it up in a field guide, I learned that this is a bird that is endemic to Western Mexico. So again, it's not in Colorado, not accurate. So that idea of putting in the include suggestions not seen nearby is not always the best idea. If for some reason you are finding that what it's suggesting isn't correct, you can always override the computer's algorithm and manually type in your identification. You don't have to go with what the computer says. You can type in a specific identification, but usually with things like birds, it's pretty accurate. Okay, so we've looked at two examples of uploading it in the upload process, but we haven't looked at what it looks like when you have uploaded the image and it's now displayed on the iNaturalist website. So this next slide is looking at the black-capped chickadee after you have uploaded it to the actual database. So you can see that I am the observer of this observation. And then you can also see that all the information that I inputted earlier, the map and the time and the date, those are all also visible on this screen. Something that is a big difference from eBird and iNaturalist here is that on iNaturalist, in order for your information to be accepted into the database, you have to have it approved by multiple people who come in, and then eventually you're given a research grade status. This is different from eBird, where you can, unless it's a rare bird for the area that you're in, you can basically put in something like say you put in a black-capped chickadee, but it was actually a mountain chickadee. That will go in the database and be accepted, and no one will ever know that it was the wrong ID and that it was accepted anyway. On iNaturalist, this is a little different, where in order for you to actually be accepted into the database, you have to get that research grade status. And we will continue to emphasize that throughout this presentation. Okay. Our next slide is going to be talking about another feature of iNaturalist, and this is the ability to be an identifier and not necessarily an observer. And you can be both, like I do both. So what exactly is an identifier? So you can see that this photo was not taken by me and not observed by me. It was observed by a different user of iNaturalist, and they uploaded a picture of a black-capped chickadee. Now, in order for this photo to be research graded, I had to come in and corroborate this ID, and then it was accepted and got the research grade status. So identifiers and observers, it's kind of weird. I know it seems like when you're an observer, you're technically an identifier because you're identifying your own stuff, but identifiers on iNaturalist really refers to identifying other people's pictures and observations. And with the next example, we are going to go a little bit more in depth about how this process actually works. So I actually did this, I added this slide just a couple hours ago. This was submitted today, August 28th, at 3 p.m. Mountain time. And so this is what an observation is going to look like before it has been research graded. I want you to note that there is a yellow box up there that says needs ID. That means that it has not yet been research graded and it needs someone to come in to either corroborate it or disagree with the identification and suggest something else. And also I want to remind you that this is not my observation, this is someone else's. So I came in earlier this afternoon, and I agreed with the suggested ID of bighorn sheep. And after that, if you go back up to the top, so that was me and me identifying it. And then if you go back up to the top, you will now see that there is a green research grade button or green research grade icon. I'm also seeing that I have two things in the chat. So I'm going to check. Okay, that doesn't look like it's super important. Okay, anyway, I'm going to keep on going right ahead. And again, if there are any questions in the chat, if y'all could let me know, that would be great.
Yeah, we do have a couple of questions.
Oh, okay, perfect.
Yeah, so Suan Hosi wants to know, I thought that iNat takes the location and time and date from the photo. I haven't had to answer those myself. So does it do that automatically?
That's a great point. So yes, it does. A lot of the times with cameras and phones, that setting is sort of already turned on, that you don't have to manually input it, which is a really nice feature because then you don't have to go painstakingly remembering like, oh, like this was at 11:54 in the morning, or oh, this is at 2:06 p.m. or something. So a lot of the time that will happen, but for sometimes it actually won't give you that data and you need to remember to put that in. That's a great question.
Yeah. And Darlene wants to know, how do you get the upload feature? How do you get to the upload feature in iNaturalist?
Yeah, okay, that's a great question also. So when you log into the iNat website, you will see I don't have a slide of this in this presentation, but you will see a huge green button in the top right corner that says upload like observations or upload images or something. It's this enormous green button. You can't miss it. And then when you click on that, you'll be able to upload photos from your computer. And then in this presentation, we will also be talking about uploading from the phone in more depth. But if you're talking about from the computer, there is this like enormous green button that's very hard to miss, and you'll just click on that and be able to upload directly from photos on your hard drive on your computer.
Thank you. That looks like it's all the questions we have.
All right, perfect. Thank you. Okay, so as you may have noticed, bighorn sheep aren't birds, and that marks sort of a huge difference between eBird and iNaturalist. You can upload anything, any living or dead organism onto iNaturalist, whereas on eBird you can only do living birds. So we're going to kind of continue with this trend. Most of the rest of this talk is not going to be about birds. There will be a couple more slides about birds, but for the time being, for much of the rest of this talk, it's going to be a lot more about non-avian organisms. So moving right along, we are going to go on to a red femered milkweed beetle. These are really common midsummer to late summer beetles here in Colorado. They're always so much fun to look for on milkweed, and kind of a fun little fact about them is that if you pick them up and kind of rub the bottom of their underbelly, they squeak. So if you ever see one, you could always try that. It's pretty high pitched though, so you have to have pretty good hearing. But yeah. Anyway, after I uploaded that photo to iNaturalist, here is what it looked like on the website. You can see my name. I'm the observer. We've gone over this before. You can see the date and time of this observation. And then finally, you can see the map of where I observed this this beetle. And the other thing I want to point out to you about this map is that you'll see a bunch of like red stick pins. Those are all other observations of red femered milkweed borers in the area. And if I were to zoom out, like hit that minus button, I would see red femered milkweed beetles for whatever area that I zoomed out to. And the frequency at which these are occurring helps me feel better about my identification, because then I know that there are other observations nearby. This seems like a pretty common species, so I feel pretty confident that I have chosen the right identification. And then of course, people come in eventually and agree with me and corroborate that ID. Okay, so we're going to move on to another kind of insect. This is an oleander aphid, which I did not immediately recognize when I was out in the field. Unlike the red femered milkweed borer that is very distinctive and that I was able to recognize right away. So we haven't really talked about how I observe my photos or my observations and how I observe organisms in the actual field. So the next image is a picture of me staring very, very, very intently at this like cluster of oleander aphids on a piece of milkweed. So I usually use my camera. I like the image quality a lot better. And if I find that it's easier to edit photos that way, and I mainly upload from the computer. So using camera is my main method of observation. Once I got those photos from my camera, edited them, and then put them up onto iNaturalist, I got it looking like this. So the lesson that I want to emphasize right here is the location and the map. I know we've talked a lot about that throughout this talk, but what I want to emphasize here is the ability to make your location as broad or as specific or private if you want. So you can see here that this is a very, very broad sort of range of where I was. You can see Lafayette's the center white dot there, but I've sort of included a lot of the rest of Boulder County, and I might even be reaching into, like, I don't know, another county, which is, you know, that works because this isn't like some kind of like super like endemic species or anything. It's nothing like that. But you can also be super super accurate with, or super precise with your location. Like here, this is actually the exact location where this milkweed plant was located. So this is like super super overboard with your identification. So you saw both ends of the spectrum there, where you can be super duper broad. You can make a huge sort of circle of where you are at. And you can also make it a super duper specific, super tiny location. There's two more options that we're going to talk about. The first one is the private locations. So you can make your location private, and in some cases, so a lot of the times people will do this if they aren't comfortable with sharing their location. And then other times, if you say found a really rare specimen of some kind of water plant, I don't know, and you don't want someone to come over here and like collect it and put it in their collection back at home. Or maybe you found a pair of nesting owls and you don't want people to go there and harass the birds. So in that case, some people will opt to use private locations. However, the problem with using private location is that your observation can never be research graded at that point. So what I would recommend instead is making a huge location, as we saw on the slide before this, so that people don't really know exactly where you were, but you give the general idea. because again, if you do a private location, your observation will never be accepted into the database for research grade status. The fourth option, which is what I use a lot, is batch uploading. I did get a question about batch uploading a couple days ago in my email, so I wanted to make sure that I included it tonight, and it is what I usually use to upload images. So a lot of times us photographers, us birders, us naturalists, we'll go out into the field and we'll take a ton of photos. Like usually I'll have 20 plus photos of a day that I spend out iNatting for, a couple hours. And it can be really, really, really annoying and cumbersome to have to go into each individual observation and select the location specifically. Again, if you have that setting on your phone or on your camera that gives you the exact location of where you are, you can bypass this entire part, makes it way easier. But for those of you who don't, there is another way to make this process a lot faster and less annoying. If you click this select all button up when you upload multiple images, and then you go over to the left and you click on the location, and then you can select a location. And when you have that select all parameter on, it will apply this location to every single one of the observations in that batch upload. So you don't have to worry about specific locations for each one, which makes the process a lot quicker. Next, we're going to be talking about another method of engaging with iNaturalist, which is engaging with iNaturalist by phone. So as I mentioned earlier, I mainly use my camera, mainly because I get better photos. I can edit them on my laptop and then I can upload them from the computer. However, you can also use iNaturalist with your phone, which a lot of people are using nowadays. So here is a picture of me in the field. I'm taking a picture of a flower, which is called a Rocky Mountain bee plant. They're super common around Colorado in the summertime. And more specifically, I'm using an app called Seek. Some of you probably don't know what Seek is. Basically, Seek is it's basically a part of iNaturalist. It is it's under the iNaturalist sort of like family branch. And the cool thing about Seek, if you guys are familiar with Merlin Bird ID, this is basically the same thing, but for taking pictures in the field, whereas Merlin is with audio and sounds. We'll get into that later though. So what you do with Seek, and what I was doing in that picture in the previous slide, is you hold up your phone to whatever you're trying to figure out what you're looking at, whatever you're taking a picture of, and Seek will actually tell you what you're looking at, which is really cool and super helpful. And it's a great way if you are in the field and you don't know what you are looking at and you want to have a better idea of what you're looking at. And usually Seek is pretty accurate, except for some, pretty obscure stuff. But for something like a Rocky Mountain bee plant, it will get that, and you can, learn stuff while you're in the field. One thing that I want to point out here is that you can post directly to iNaturalist from Seek. This is a feature that I don't personally use very often because I do find that the Seek camera is not super reliable, and it's usually a little bit out of focus. So I usually use Seek merely for figuring out what I'm looking at or trying to ID something that I'm not familiar with. If I happen to be uploading from my phone, which I occasionally do, most of the time I'm using my camera and my laptop, but sometimes I will take pictures with my phone. You can use the iNaturalist app. So I want to stress that this is different. This is a different app than the Seek app, but Seek and iNaturalist are both part of iNaturalist. So you can see that the iNaturalist app is very similar to the desktop layout. of the iNaturalist app. The first thing you do, you fill in those parameters of location, date, and time. For my phone, I don't have my location turned on, so I actually do have to fill in the location. You can just search and put in a specific place of where you are at. So once I had that filled in, that's a pretty broad location. The next thing you do after you have your location and your date and your time, like with the uploading on the computer, you're gonna go next to actually figuring out what you're looking at. So you can see that it suggests, based on the parameters that I put in with location and date and time, that it suggests a Rocky Mountain bee plant. It suggests the genus to which the Rocky Mountain bee plant belongs. And it also suggests 3 other similar looking flowers. So I proceeded to select Rocky Mountain bee plant. I did this for 2 reasons. The first one, I'm very familiar with this plant. It's all around Colorado. It practically grows in my backyard. I see it everywhere. And then also, I had seeked it only a couple minutes earlier. And it had given me that idea of Rocky Mountain bee plant. So I was pretty confident that what I was looking at was indeed a Rocky Mountain bee plant. Okay. We are going to now continue on with the presentation with a spotted spread wing, which is a kind of ode. We're kind of in an ode theme for the next few slides. We have quite a lot of odes coming up. This one in particular, I found quite beautiful, and it was being very photogenic for us. It was sitting on this, or I guess it was perched on this milkweed leaf. It wasn't moving. We were able to get really close to it for some good macro shots of it. So after I took this picture, again, I edited it, all of that stuff, and then I uploaded it to iNaturalist. So this is what it looked like on the display, and the particular lesson that I want to point out for this observation is the fact that there are 4 images here. Some of my other observations that I've showed in the past slides did also have multiple photos, but I didn't really talk about it until now. So one of the really cool and one of the really valuable things about iNaturalist is that you can upload multiple different photos of the same organism within the same observation. This can be really helpful for 2 reasons mainly. The first one, more scientifically, is that if you are identifying a particular organism that needs multiple photos, multiple angles, an example would be in butterflies, when a lot of the times you need a picture of the outside and the inside of their wings in order to get an ID for some of the harder butterflies. And for a lot of times for things like damselflies and other odes, which I've learned through uploading a lot of photos of these to iNaturalist, is that a lot of times experts need to be able to see like a side profile, a top, and then like from the back or from the head. So it's really valuable a lot of the time to upload multiple photos of the same individual. Another thing, if you have a lot of great photos that you want to share online with people for people to look through, you can always upload a bunch of your favorite photos. The other thing that is a little different about this observation that I want to point out is the fact that when I uploaded this, I didn't get it right down to species level right away. I knew that this belonged to the genus Lestes. I've seen other spreadwings like this, so I was relatively familiar with the general identification of it, but I didn't know specifically what species this belonged to. So I put in the genus, and then people who are experts at identifying this specific genus came in within the next few days and were able to tell me what I was looking at specifically. So again, I had the genus here initially, and then later 3 other people came in and were able to give me an exact actual species of what I had been looking at. And then with those 3 identifications at species level, I was able to get to the research grade status. Okay, so we'll have about 5 more minutes until our break. I want to check, are there any questions right now, or should I keep on going?
Currently, there are no questions.
Okay, perfect. Again, we have about 5 minutes, so I'll cut us off for a break in about 5 minutes. Our next ode, like I said, we will be spending a lot of time with odes for the next few slides. This is a plains forktail, which can look similar to the last ode we just looked at, the spotted spreadwing, but they're actually completely different species and they're not in the same genus either. So here is what this looks like when I put it onto the iNat database. Again, it's very similar to what we've seen in the past slides. And I want to emphasize here that unlike the other ode, I knew right off the bat what exact species this was. Eventually, some people came in and corroborated my ID, and it got that research grade status. Moving right along to our last bit of odes. I promise we'll be done with odes after this particular lesson. I love them though. So these are 2 band-winged meadowhawks. Meadowhawks are a pretty common dragonfly here around Colorado, and these are 2 different individuals of the same species that we observed last Saturday. Okay, so here is a side-by-side comparison of those 2 observations that we had last weekend. You can see here that I put in bandwing meadowhawk for the yellow one because I was familiar with this one. This is a really common dragonfly here around Colorado in the summer and especially in late summer and early fall. However, for the other one, the red dragonfly, I actually didn't know what it was right away. And I put in meadowhawks. I knew it was a meadowhawk, but I actually didn't know what species it was. So I was pretty surprised when the experts came in later and told me that this is actually also a bandwing meadowhawk. So I think that this is just like one of those lessons where it's always important to upload different individuals of the same species for scientific purposes. It also helps the iNat algorithm become a better identifier of whatever you're looking at, and it also helps you become a better scientist in general. I was not familiar with the red color of the bandwing meadowhawk, and I was pretty excited to learn that that is actually also a bandwing meadowhawk, and I learned that. And also I want to point out that as far as I know, the different colors don't mean any differences in sex or in age or anything like that. I don't actually know what causes those different colors, but I was pretty happy to learn that there are different patterns for different individuals within the bandwing meadowhawk species. Okay. We are now going to move on to a fly, which I observed at last weekend on Saturday. I saw this particular fly and it was really, really small on a piece of milkweed. Milkweed has so many incredible insects. I mean, I think it's probably most famous for the monarchs and the caterpillars, the monarch caterpillars on the milkweed, but there is a ton of other cool stuff that you can see milkweed. So if you're really into insects like I am, always look at milkweed because there's always cool stuff there. This one in particular, I was very struck by the iridescence of the body of this fly, as well as the speckled kind of brownish pattern on the wing. It kind of reminded me of a cow. Anyway, I thought that this was a really, really beautiful fly, and I didn't know exactly what it was. So naturally, I put it up onto the iNat database. So here you can see that again, like I mentioned earlier, I knew it was a fly. I didn't know exactly what it was for species, so I put in a pretty broad ID, sort of like I did with spread wings, but this one is even broader. I put in a subfamily. Um, and then because I wanted to learn more about this fly and know what it was, know more specifically what it was, if possible, I tagged two experts, two people who are top identifiers of this particular subfamily. So it is 6:45 now. I know I'm in the middle of a slide right now. However, I will come back to this in just about 5 minutes. I'm going to turn off my audio for the 5 minutes. I will turn that back on when I come back in 5 minutes, and we will continue talking about this fly. So I will see y'all in 5 minutes. Alrighty, it is now 6:50, and I want to make sure, can people hear me? Just make
Yes, I can hear you. And there
perfect.
are questions in the Q&A.
Yes, I can see that. And I am having a weird thing on my laptop where I can't move the question and answer. So I can see there's 4, but I can only actually read 3 of them. So I will read the 3 that I can see and answer them right now, but I can't see the 4th one. So once I get there, if you could read that one to me, that'd be great.
Yeah.
So the first one is from Barbara, and it says, once you get a species ID, do you change your ID? So what I'm assuming you're asking is that when you put in a species, if it ends up being wrong, or if it ends up being wrong, can you change it? And the answer is yes. You can always change your identification that you put initially in, and a lot of times, if maybe you identify it wrong, someone else can come in and correctly identify it for you. We have another question after that from Kimball that says, basically, a lot of the time people have super silly usernames for iNaturalist, and how can we have a more, how can we get the observer to have a more formal name that's a required part of a bird record data point? And this is a great point. I 100% agree with you that it can be very frustrating if someone has a super silly name. I started off iNaturalist with a name of Cheetah Fanatic _29. That was my name for like a year. I only recently changed that to Hannah Floyd _naturalist, but I completely agree with you that it is super frustrating when people don't have those names. And the usually the fix for this, to get to have the name for a bird record, is if you click on people's profiles, more times than not, in their profile, they're going to have their actual name there listed underneath their username. So usually you'll be able to see it there. It is an extra step, but usually it'll be right there. And if all else fails, there is a way to like chat people on iNaturalist, or you can always add a comment like you mentioned. Okay, our next question is from Pamela, and she asks if the app can be downloaded to phone and or laptop at home, and can Seek be used on either if you have the photo. So the app, there's a phone app for both Android and iPhone users. And the app on iNaturalist Online is not really an app, it's a website. So you don't really download it on your computer at home. It's just something you look up. But on your phone, you do have to download it. And Seek can only be used on your phone. You can only, it's an app that's only on your phone. It's not online. So for using Seek on the computer, that doesn't exist. That's not an actual function of Seek. And then our final question is, how do you find the appropriate expert to tag on your observations? This is a great question, and I will actually be answering that on this slide with the fly. So hang tight. I know that I haven't talked about that yet. I have done that intentionally, and we will learn about that on this slide. So thank you for all of the questions. We are going to move right along now and continue with where we left off. And I'm going to make sure that I can move the slide. Let's see. Okay, that worked. Okay, so as you can see here, I have tagged 2 different people. And one of them came in and was able to narrow down my identification to the genus level, which is much better than what I had at subfamily. So, like the question that was asked at the end, how in the world did I know how to do that, or what experts to tag? And that all comes from this. So far with our looking at the desktop, we haven't been able to see what is below the screen captures I provided. So if you were to be on the desktop and scroll down just a little bit, you would see this sort of like a little box that says top identifiers of blank. And the blank is whatever species, whatever family, whatever genus of organism that you have observed. And it's going to show you the top identifiers, usually experts in their fields, for that particular organism. So here you can see that when I tagged those 2 people, I found them from this section. I knew that these 2 people were reliable identifiers, so I tagged them, and sure enough, one of them came in and was able to give me a much better idea of what I was looking at. And we'll continue to kind of explore this and emphasize this in some other examples in the future. The next thing I want to talk about here is the project sort of icon. We haven't talked about this yet either. Projects are a cool feature of iNaturalist. They are basically either they can be used for taxons, they can be used for geography, they can be people, interest groups that, but what they're doing is they're different research sort of groups. And again, like I said earlier, they can be based on geography, as is this one for Boulder County. So anything that you observe that's wildlife in Boulder County will be added to this project once you get added to the project. Or it can be geography, so like birds of the world, slime molds of the world, whatever you want. Or it could be people. So for example, I'm also part of like a young birder, young naturalist group on iNaturalist. And the purpose of these projects is to document information in the specific parameters, in the specific bounds of what the project is. And for this one specifically, I'm part of Boulder County Wildlife. You can see me down there. I am catching up to Steve Malad now, which some of you may know, probably know, and I am slowly creeping my way up the leaderboard for Boulder County Wildlife. And this project, by the way, is sponsored by Boulder County Parks and Open Space. And so a lot of these projects will be sponsored by actual like research entities and organizations. Our next slide is going to be about fish. Fish, obviously, I'm not an expert at these at all. I don't know anything about fish. But I saw these when I was at the lake right next to my house, and I was particularly curious about the red kind of colored fish in the background. I did not recognize that fish immediately, and I uploaded it. Again, I wasn't very sure exactly what this was, so I put in my identification of ray fin fishes, which is basically all the fishes here in Colorado. And by the way, that encompasses a huge number of species. So like, you know, birds is a class, ray fin fishes is also a class. So if you imagine all the birds of the world, it's kind of like that, but for fish. And I also made sure to note that I'm talking about specifically the red fish in the background. And this is a pretty important comment to put here because it's not always clear what observation that what organism in an image you are looking at or looking to get identified. For example, if you had a sunflower, let's say, with an American goldfinch eating some of the seeds on it, and you just put in the observation without an identification, people wouldn't know if you were looking at the sunflower or if you were looking at the bird, or maybe you're looking at a tiny little leaf hopper on the plant. So it's always important if you have more than one organism to note which one you're looking at, as is here. So anyway, you can see that I, asked what specifically was the red one. Are these two different species? I didn't know. And so again, I tagged 2 people. I got that from the bottom of the where we were looking at the last example of the top identifiers of rayfin fishes in the world. And the first one said, hey, sorry, I don't know freshwater fish. Cool. And then actually another guy came in and he gave me an ID for both of the fish. And I learned that both of the fish are actually completely different species. So the one that I had put in for this observation specifically is called a green sunfish. And then the other one, which I hadn't yet had an observation for because I didn't know what it was exactly. And I wanted to know before I uploaded a picture of it, was a largemouth bass that they put in, and they told me about that. And then I ended up subsequently uploading that to iNaturalist later that day. So next up, we're going to move on from our fish, and we're going to talk about dead organisms. So this also marks a difference from eBird. You can upload dead organisms onto iNaturalist as is here. There's actually 2 dead organisms. I didn't notice the tiny oleander aphid that we were looking at earlier, but the main thing that I uploaded this for was for the European paper wasp. So I uploaded the European paper wasp, and as I noted before, it is evidently deceased. And I came in and added an annotation in the annotation field. This is another aspect of iNaturalist that I wanted to bring up. You can add specific annotations of your organism. So this can include the life stage, the sex of your organism, here alive or dead. So in here I put dead just for more information for this particular observation. The other thing that I want to point out here is this pink check mark, or not check mark, pink exclamation point, which we haven't seen yet. For different organisms and different observations, iNat will put in these special statuses. So for this one, this is a particular organism that has been introduced into Boulder County via anthropogenic means. And then there are also other statuses that iNaturalist can provide. For example, if something is endemic or if something is critically endangered, it will provide those, and they're all different symbols at the top for you. We are now going to move on to a bird feather. So again, on eBird, you can't upload a picture of a bird feather, but on iNaturalist, it is totally allowed. You are allowed to upload parts of organisms, as is here. So here is what I uploaded to iNaturalist. I wasn't entirely sure what it was when I uploaded it, so I put in just birds as my ID. But now looking back at it, I do think that it is a primary feather of the common grackle that we mentioned in the beginning from our eBird checklist. And you can also see another example of an annotation here, which is the evidence of presence, which is yet another field that you can fill in here. So this could include, for example, feather, as is here, or it could be scat. If you take a picture of bear scat or coyote scat or something like that, this could be footprints, like raccoon, like handprints, or like the turkey tracks or something. And it could be fur, it could be any number of things. For this one specifically, it was a feather. Okay, so that was just a bird after a long string of non-avian organisms. We are going to continue with this bird trend with our revisiting of this gray catbird. So The gray catbird here, this is a photo. This is also taken by my dad while I was looking at hummingbirds. So obviously this is a photo. It's totally able to upload this to iNaturalist, totally allowed. However, this next example is a video. Currently, it's probably going to change eventually. You can't upload videos to iNaturalist. Like I said, you can upload videos as we saw to eBird, as we saw in the beginning with that eBird checklist where it had an audio, video, and photo capability. But I want to stress, you can't upload videos to iNaturalist. However, I do think that's going to change in the future. And another media that we took of this bird, of this gray catbird, was a sound spectrogram, an audio recording. So my dad, again, took this audio, and you can see here that those are the catbird's actual sounds calling. If you went to Nathan Peeplo's talk about identifying, looking at spectrograms on, I think it was Friday night, you'll probably learn all about these, but those little bars are going to indicate that it's a nasal sounding call, which matches the gray catbird. You'll probably also notice that there is this sort of gray fuzzy bar at the top, and those are crickets. And those are not birds, obviously, but we will be coming back to that in a short while. And then also the rest of the spectrogram is sort of fuzzy. It's kind of gray and fuzzy, and that is ambient noise. And that is kind of expected in a lot of your typical recordings. So that's like the water feature that the gray catbird was actually frolicking around in. That's footprint or footsteps, people talking, grass crunching, leaves rustling, whatever. You can't escape it when you're out in a public park. And the thing that I want to stress here is that audio is acceptable to upload to iNaturalist. So videos can't upload to iNaturalist. However, photos and audio are, you can upload those to iNaturalist, and we'll be talking a little bit more about audio on iNaturalist in a bit. I do see a question that came into the chat. It says, is iNaturalist only for photographers? That would be an enormous difference from eBird. The answer is no. A lot of non-photographers upload pictures to iNaturalist as well. It's also as much of a scientific database as eBird is. A lot of the time, actually, tonight I've tried my best to show you good images, but a lot of the time on iNaturalist, you're going to have images that are of terrible quality and you can't really identify it. So no, iNaturalist is not only for photographers. There's definitely people that upload their like prettiest and best photos. I always try to upload my best photos, but of course, if I'm looking at something that is rare and unexpected, then I will upload those photos even if they're not of the best quality. So you don't have to be a professional photographer to use iNaturalist. You can be a whatever photographer and still get a lot out of it. Great question. We're going to shift just for a brief little bit to Merlin Bird ID, which is part of eBird, and it was released earlier. The capability to identify birds by audio, by calling, by song was released earlier this year. And it is very, very accurate, as you can see here, where it heard the catbird in when we were out in the field, and it was able to get it right away. So with Merlin, again, I'm not going to go super into this because this is not about Merlin Bird ID, but you can hold up your phone when you're out in the field, have it identify it for you. For us specifically, we uploaded our sound spectrogram. As you can see here, this is the same sound spectrogram from the previous slides. Those two bars are the catbirds, and then still that long bar at the top, those are still the crickets. And I promise we're going to get to the crickets in just a little bit. But that is Merlin Bird ID. And the main thing that I wanted to point out about this is that Merlin Bird ID can identify whatever you are observing by sound. It can do it right away. iNaturalist can't do that. So while you can put audio on iNaturalist, it can't actually tell you what you're hearing. This is another function of iNaturalist, or dysfunction rather, that I think is going to be fixed and updated in the near future. So as of right now, if I were to put this gray cappered audio onto iNaturalist, it actually wouldn't be able to tell me what I'm listening to. I would have to manually put in what I think it is, and then people will come in later and ID it for me as well. So finally, we're going to get to the crickets. I know everyone is itching to get to the crickets. Here is a audio, or a spectrogram of the crickets that we have been talking about now for many minutes. Again, if you were at Nathan's talk a couple nights ago, you're probably very familiar with these spectrograms. This is a sound recording of the crickets, and you can see in the bottom one, this is the actual spectrogram, and you can tell that it has a pretty high frequency. It's at around 9,000 hertz, so very high frequency. And then the top part of this is called an oscillogram. This is getting a little bit technical, but basically an oscillogram just measures how loud whatever organism you are looking at. And this can be quite helpful for insect ID, especially measuring the loudness of whatever you are listening to. So we're going to now get on to the uploading audio to iNaturalist, because as I mentioned earlier, you can't, the iNat algorithm can't tell you what audio you're listening to from the very beginning. You have to manually put that in yourself. So this is an example of an observation that my dad put into iNaturalist last Saturday. He put this in as an Allard's ground cricket, and this is the recording that we have been looking at, the crickets that have been mentioned a lot in the last few slides. So my dad, as you can see, suggested this ID. Again, this was not the iNat algorithm's doing. This was his own doing. He has experience and some knowledge with identifying these because he spent a lot of time learning about identifying Allard's ground crickets. However, he wanted to be sure, so he went down to the bottom corner and tagged 2 people he knew would be able to identify the crickets for him. And later that day, someone came in, Brandon Wu, who is an orthopteran expert on iNaturalist. If you get into iNaturalist and do a lot of grasshoppers, I 100% guarantee you'll run into him eventually. And he came in, and I agreed with my dad's ID, corroborated it, and it was accepted into the iNat database. Again, I want to emphasize that this all, here's the comments. Sorry, I can't see part of my screen right now. So, but as I was saying earlier, those are the parts where my dad wrote to someone to ask them for confirmation of what they were looking at, and then subsequently that confirmation came later that week. So that comes from down below there, where it tells you the top identifiers in the world for Allard's ground crickets. And this is all mainly because, again, iNaturalist can't identify your audio specifically right when you put it in. You have to put that in manually. So it's always a good idea, if you upload audio, to ask someone to tag someone to see if what you're seeing, to see if what you have observed is actually correct and your identification is correct. And again, hopefully iNaturalist will fix this in the future. I have a feeling that this is a capability that will be incorporated into iNaturalist in the near future. Okay, so Brandon Wu, obviously, and some of the other people we've been seeing on those slides are top identifiers in their fields. Throughout my time on iNaturalist, I have also built myself up onto the ranks for some relatively more easy things to identify and things that I'm more familiar with. So you can see me there for the top identifier of blue fungus beetles. These are a really, really cool beetle that we have here in Colorado. They like a lot of rotting, moist wood in the foothills of Colorado. And I've seen them a lot. I have a lot of experience. Sorry, I have a lot of experience with them, and I feel pretty confident with my identification of them. So I have done a lot of identification of blue fungus beetles. And I've also started identifying a lot of other stuff, as you will see with this next slide. Um, this is my profile page. And I know there was a question earlier, I think it was from Kimball, that asked about how we can see people's actual name. So you can see there that my name, Hannah Floyd, is displayed right there. And a lot of times people with those really crazy, wacky names like me, who was Cheetah Fanatic _29 for a long time, my name, Hannah Floyd, was still there. So people could still actually know who I was, but not from my username, which was a little ridiculous. But here you can see that it provides sort of the stats of your time here on iNaturalist. I have, just around 3,600 observations. I have 1,300 species, and then I have 13,000 identifications. And there's a bunch of other stuff down there, but the main thing I want to point out to you there is the identifications, and that my time here on iNaturalist, I've actually learned just as much from identifying other people's stuff as I have from observing my own stuff. A lot of the times with things that you're really familiar with, so like a lot of birds here around Colorado, some plants, I've started familiarizing myself with a lot of herps, a lot of oats around Colorado. So I started making a lot of these identifications here in Colorado, and it's actually helped me become a better naturalist. So with all of that being said, I am basically done here. I have one last big slide, and that is talking about the community on iNaturalist. I have not only met a ton of scientists, experts in their field on iNaturalist, but I've also bonded with other people who love observing stuff as much as I have. I have gone on a lot of trips recently. With the pandemic, I have been able to get outside a lot more and see a lot of people and do all this iNatting, and it's really, really cool. And it's been really, really cool to experience all that with all these people. So these are some of my favorite memories from the past 2 years. The top left corner, that was this summer. I co-led a tour of the San Luis Valley here in Colorado. And this was a particularly cool day where we went up into this sort of montane area, and we had some great looks at Hammond's flycatchers. We had a beautiful warbling vireo perched right out in the open for us. We had some amazing butterflies and some really cool lizards. The next photo in the middle right there, that is from the Snow Goose Festival in February of 2020. That particular day, it was freezing cold, but we braved the cold weather and we got stunning looks at horned larks and Lapland longspurs. And even though it was freezing, we were all so cold. It was a really, really memorable day. The next photo is from this past summer, actually. It was one of my most sought after herps that I had been wanting to see for a really long time. Through iNaturalist, I've gotten really into herps, and I wanted to learn so much more about herps and see them all the time. So we had some friends who were visiting, and we went up to Pawnee National Grassland, which is up near Fort Collins in Colorado. And what I'm holding in my hand, and that's my dad and myself photographing it, that is a greater short-horned lizard. And they kind of just scamper across the grasslands out there up at Pawnee. And it was a really special experience to see that. The bottom left corner, that was just last week, I think. We had some friends visiting from Missouri. We went up and we had a great day for butterflying and iNatting in general. There we were looking at a particular butterfly called a wood nymph. They're really quite beautiful butterflies. Middle bottom, you might recognize those bird feeders actually. This was taken in the summer of 2020, I believe, and those are the two kids that I babysit. I have the world's best babysitting gig because the two kids I babysit are bird watchers. So when I babysit them, I just drive them to places and we go birding and iNatting, and they love it. So here in particular, they were observing hummingbirds up close at the local feeders. And finally, our last image, this was also taken earlier this summer. I think it was July. That is me and another friend visiting Colorado. We took her up to subalpine tundra here in Colorado, and we were specifically looking for snowshoe hares, which are very elusive mammals up here in Colorado, and I'd never seen one before. We actually did end up finding one, which I was very excited about. And in addition to finding that little, really, small rabbit or hare, not a rabbit, a hare, we also found a moose. As we were driving around a corner, there was a moose in the middle of the road. So those are particular memories that I have from iNaturalist. Again, iNaturalist is not all, a big part of it is about science and contributing to research, but it's also about meeting new people. There's a community there. Everyone is so helpful there, and it's a great way to meet other people. I see a question in the chat from, I think, Lena is how you pronounce it. And it says, I'm actually going over your question right now. So it says, when you record bird sounds in the field, how do you upload onto eBird? Do you need an app to change into a spectrogram? So when we record bird sounds in the field, we do it there's two ways that I know of off the top of my head. I'm actually not very well versed with using spectrograms. That's mainly my dad. But the two ways that when I do use spectrograms, my dad actually has a recorder and like sort of sound microphone that we can hold up to whatever we're recording. Usually it's some kind of bird or a cicada or a cricket or something. And later we go onto our computer and we mess around with the background noise, making what we're actually trying to be heard a lot louder and changing all of that. Another way that you can do it is using, a lot of phones have sort of like sound files. I don't quite know what, I have my phone right here. Voice memos, that's the word I was looking for. Voice memos is an app that I know Apple phones have, and I'm sure Android has an equivalent of that. But basically, you can use that to record things, and then you can actually upload from those to your computer to iNaturalist, which I have done a few times with coyotes specifically. I have a specific memory of doing that with coyotes. Okay.
Hannah, there is another question that came in through.
Oh, perfect.
To me.
Okay.
John Harris would like to know if you wanted to be notified when somebody reports a blue fungus beetle, could you set it up to do that?
So I'm assuming, so on eBird, obviously there are the eBird rare bird alerts, which I get those emails every single day of rare bird alerts. For iNaturalist, as far as to my knowledge, you can't actually set notifications of when specific observations for specific species are made. Usually the way to do it is that if you use iNaturalist all the time like me, you kind of go in and check what you're an expert at. And once you get to that point where you're like a top identifier for a certain taxon, people will tag you regularly when they observe those. So you'll get comments. And again, we didn't talk about this in this presentation, but similar to how someone earlier asked where the observe button is, how you upload it on the website, there's a little comment icon at the top. And when you click on that, you'll be able to see all the people that have tagged you for help. But there's not really a specific way to set a notification for a specific species or taxon that you want to be notified of when it's observed. Okay. I'm going to move along to basically the final slide. This is my acknowledgement slide. I have three main people to thank. It's been a long, long presentation. So I want to thank Jennifer Raisenga, Frank Izagiri, and my dad, of course, for guidance along the way with all things iNaturalist. Jennifer Raisenga and Frank Izagiri are two iNat giants. They have so many observations and identifications on iNaturalist, and they really inspired me along the way to start my own iNaturalist account. Also, I want to thank Western field ornithologists for providing so many opportunities to me and my family over the years, including with this past workshop and the presentation that I gave last Thursday, I think it was. And so that concludes my presentation. That concludes this workshop. I have Q&A time. We're basically at the time limit. We have about 5 more minutes. So I don't see any other questions right now. If you have anything, please write it in the chat now. But as of right now, I am done. Thank you so much for everyone to everyone for coming out here tonight. I hope you learned something. And good night.
Thank you, Hannah. That was an awesome presentation.
Thank you.
Last summer, during the pandemic, I managed to get up into Yosemite National Park a few times, and I was photographing butterflies there. And I went to a trailhead, and there was a sign posted by the rangers, and they had a project, basically just as you described, where they were asking people to report pollinators in the meadow at Lucan's Lake to their pollinator project. And that's what I did. And it was really a great thing. And one of the things that I noticed was when I posted one of my butterfly photos, within a few hours, I had a response from somebody who was actually doing research on that species. And it really was amazing. So I really appreciate this presentation, and thank you very, very much.
Thank you for having me.
Our next presentation. You bet. We're so grateful. We'll take a brief break, and then our last program of the day today is Nathan Peplow's Sound ID Challenge. So we'll see you all soon.